One of the most essential aspects to protecting against any virus or disease is the methods in which you diagnose patients. And we've seen this with the COVID-19 pandemic. Countries that are diagnosing their patients more often are likely to be doing better than other countries that aren't putting as much effort into diagnosing patients. And one of the most important aspects to ensuring you have a good diagnostic method is the time it takes and the accuracy. And we've seen with RT-PCR, reverse transcription polymerase chain reaction, that it's not meant for the year we're living in right now. This is the exact reason Jibraan and I are going to propose to use CRISPR-Cas13, not only as a diagnostic tool, making it much more quicker than PCR while also keeping that same accuracy, but also using it as a therapeutic, potentially saving hundreds of millions of lives for people who are struggling with COVID-19 and don't have the vaccination yet, but also making it really easy to adapt to different viruses. So I'm first going to be talking about RT-PCR and how exactly it works. So the first step to reverse transcription polymerase chain reaction is actually obtaining the sample. And after you obtain the sample, you're then going to reverse transcribe that sample that contains RNA into DNA. And this is really important because COVID-19 is an RNA-based virus, and in order for PCR to actually work, you're going to have to reverse transcribe that RNA into DNA. And after the RNA is reverse transcribed into DNA, you're going to want to amplify the DNA using polymerase.
Awesome introduction, Ali. But before we get started about how CRISPR is going to tackle COVID-19, we need to first understand that everything around us is made up of DNA. You see, DNA is the fundamental building block of life. Everything is encoded by DNA, and DNA itself is made up of just 4 letters: A, C, T, and G. This code is read by biological machines within our cells, and depending on the sequence of the code, it can produce various proteins. Now, let's say in the event of a letter change, also known as a mutation, an abnormal protein can be formed. This is the case in sickle cell anemia, where a mutation takes place in red blood cells. This is also where CRISPR comes in. Originally discovered in bacteria, CRISPR has the ability to locate and cut certain DNA or RNA strands. In other words, CRISPR is like a find and cut tool, but for DNA and RNA. Now, the way scientists actually guide CRISPR to cut things is through something called a guide RNA. A guide RNA is a piece of RNA that matches the target DNA sequence that has to be cut. The guide RNA essentially guides the Cas9 enzyme to that spot. You can think of a guide RNA as the map or the instruction manual for Cas9. For this video though, we're going to be focusing specifically on CRISPR-Cas13, which cuts RNA, DNA's cousin.
So the real question is, how is CRISPR-Cas13, a mechanism that's usually used for gene editing, going to be able to be used for diagnosing COVID-19? Well, the method that CRISPR-Cas13 is going to function in is a lot more efficient than the PCR method. That's because once CRISPR-Cas13 finds the COVID strand that it's looking for, it's going to slice all the RNA around it. And because it slices the RNA, scientists have put in certain reporters that when sliced are going to give a visible signal. This visible signal is going to allow people to understand whether a patient has COVID-19 or not.
COVID-19 is caused by an RNA virus known as SARS-CoV-2. Now, the reason I mentioned CRISPR-Cas13 is because of its ability to cut RNA. Now, scientists at Stanford University designed a system called PACMAN. Yes, like the video game, that stands for Prophylactic Antiviral CRISPR in Human Cells. What does that mean? Essentially, it's just a fancy term for CRISPR-Cas13 mixed with a bunch of guide RNAs. Now, the whole idea behind PACMAN is to target and cut viral RNA. If this viral RNA is cut, SARS-CoV-2 won't be able to replicate, and therefore the problem is solved. Like I said, CRISPR-Cas13 uses guide RNAs to target and locate certain strands. These same scientists found that just 6 different guide RNAs can target 90% of all coronaviruses known to us, and just 22 can target 100% of coronaviruses. So as awesome as PACMAN is, there is still a long way ahead. Honestly, we don't really know that much about CRISPR. In fact, CRISPR originally were discovered in bacteria, specifically bacteria that are pathogenic to us. So that as well brings up a concern about whether or not CRISPR proteins will trigger an immune response in our cells. One of the biggest issues is delivery. Specifically, how do we know which cells are infected and which cells are not infected? And how do we even get CRISPR into our body and into these cells? One way is with adeno-associated viral vectors, but they're technically viruses too, and that brings up immune response concerns. However, scientists are currently looking into lipid nanoparticle technology to deliver the CRISPR components. And to top it all off, CRISPR has been known to cause off-target effects, that is, cutting in places other than the target sequence. This is because CRISPR-Cas proteins will cut a sequence even if it slightly matches the guide RNA sequence.
The potential that CRISPR has to be used as a therapeutic for viruses coming up in the world today is truly endless. It is so easy and quick to adapt the CRISPR mechanism to any virus so that it can be used as a therapeutic and hopefully defend against that virus for years to come. The craziest part is this is only 10 to 20 years away. We're able to completely change the way we fight against diseases with the use of CRISPR-Cas13, CRISPR-Cas12, or potentially even CRISPR-Cas9 as a therapeutic to viruses.